Welcome to this scientific lecture on healthy food habits and their remarkable role in DNA repair. Today we're going to explore how specific nutrients and compounds in our diet can actually help repair the very blueprint of life, our DNA molecules. First, let's understand what we're dealing with. Every single day, each cell in your body experiences tens of thousands of DNA lesions. That's damage to the genetic code that makes you who you are. Here's a representation of a single cell. Those yellow dots represent sites of DNA damage occurring right now. This damage comes from multiple sources. What causes this damage? The sources are both external and internal. First, reactive oxygen species, or ROS, are produced during normal cellular metabolism. These are highly reactive molecules that can attack DNA. Second, ultraviolet radiation from sunlight can cause thymine dimers and other photoproducts in DNA. Third, environmental toxins, industrial pollutants, and certain chemicals can directly damage DNA structure. Fourth, even the normal process of DNA replication isn't perfect. Errors occur at a rate of about 1 per billion base pairs copied. Now here's the remarkable part. Your body has evolved sophisticated DNA repair mechanisms. These are molecular machines that patrol your genome, find damage, and fix it. Imagine repair enzymes as molecular mechanics. They scan the DNA strand, identify damaged sections shown by this red star, and initiate repair. But here's the critical question we need to address. What do these repair systems need to function optimally? The answer is specific nutrients from our diet. Let's explore the key nutrients that have been scientifically demonstrated to enhance DNA repair mechanisms. First, let's discuss folate and the B vitamin family. These are absolutely critical for DNA synthesis and repair. Folate is converted to tetrahydrofolate, which is essential for synthesizing nucleotides, the building blocks of DNA. When folate is deficient, uracil gets misincorporated into DNA instead of thymine. This creates sites that must be repaired, increasing the burden on repair systems. What foods are rich in folate? Let me give you specific, evidence-based sources. Leafy green vegetables are exceptional sources. 1 cup of cooked spinach provides about 260 micrograms of folate. Legumes are even better. 1 cup of cooked lentils provides 358 micrograms, which is nearly 90% of the daily recommended intake. Cruciferous vegetables like broccoli and Brussels sprouts are also excellent sources, providing around 100 micrograms per cup. The second category is antioxidants. These molecules neutralize reactive oxygen species before they can damage DNA. Let's start with vitamin C, also known as ascorbic acid. This is the primary water-soluble antioxidant in cells. Here's how it works. Vitamin C donates electrons to reactive oxygen species, neutralizing them before they can attack DNA. The reaction is, vitamin C plus a reactive oxygen species yields oxidized vitamin C plus water. The threat is eliminated. Which foods provide therapeutic levels of vitamin C? Citrus fruits are well known. 1 medium orange provides about 70 milligrams of vitamin C. But here's a surprise. Red bell peppers are even better. 1 cup of chopped red pepper provides 190 milligrams. Kiwi fruit is another excellent source, with 1 fruit providing 64 milligrams. Now let's discuss vitamin E, which is fat-soluble and protects cell membranes from oxidative damage. Vitamin E prevents lipid peroxidation, which is the oxidative degradation of membrane lipids. This is critical because damaged membranes can lead to DNA damage. What are the best food sources? Nuts and seeds are exceptional. 1 ounce of almonds provides 7.3 milligrams of vitamin E, which is nearly half the daily requirement. Sunflower seeds are even better, providing 10 milligrams per ounce. The third category is polyphenols and phytochemicals. These plant compounds have remarkable DNA protective effects. Let's focus on sulforaphane, which is found in cruciferous vegetables like broccoli. Sulforaphane activates the NRF2 pathway, which is a master regulator of antioxidant response. This upregulates dozens of protective genes. The pathway is sulforaphane leads to NRF2 activation, which leads to expression of antioxidant genes. Research indicates that 3 to 5 servings of cruciferous vegetables per week provides optimal DNA protection. The best sources are broccoli sprouts are extraordinary, containing up to 100 times more sulforaphane than mature broccoli. Brussels sprouts and cabbage are also excellent sources. Now let's discuss resveratrol, a polyphenol that activates sirtuins, which are proteins involved in DNA repair. Resveratrol activates SIRT1, a sirtuin that enhances DNA repair and genomic stability. Resveratrol is found in red grapes, blueberries, and dark chocolate with high cocoa content. The fourth category is omega-3 fatty acids. These are essential for maintaining DNA integrity. Omega-3 fatty acids, particularly EPA and DHA, reduce inflammation and oxidative stress, both of which cause DNA damage. The mechanism is omega-3 intake leads to anti-inflammatory effects, which leads to less DNA damage. What are the best dietary sources? Fatty fish are the gold standard. A 3-ounce serving of salmon provides about 2.2 grams of omega-3s. For plant sources, walnuts are exceptional. A quarter cup provides 2.7 grams of alpha-linolenic acid. Flax seeds and chia seeds are also excellent plant-based sources. The fifth category is trace minerals that serve as cofactors for DNA repair enzymes. Zinc is absolutely critical. It's a cofactor for DNA polymerase and many repair enzymes. Studies show that zinc deficiency can impair DNA repair capacity by up to 50%. This is a massive effect. The best dietary sources are: oysters are the richest source. 6 medium oysters provide 32 milligrams of zinc. Red meat is also excellent, with the 3-ounce serving providing 7 milligrams. For plant sources, pumpkin seeds provide 2.5 milligrams per quarter cup. Selenium is another critical trace mineral. It's a component of glutathione peroxidase, a powerful antioxidant enzyme. Selenium enhances glutathione peroxidase activity, which neutralizes hydrogen peroxide and other reactive oxygen species. Brazil nuts are extraordinary. Just 1 ounce provides 544 micrograms of selenium, which is nearly 10 times the daily requirement. Now let me address an important question. Can we repair all DNA damage through diet alone? The answer is no. Diet alone cannot repair all DNA damage. However, proper nutrition significantly enhances your body's endogenous repair capacity. Diet provides 2 critical things: the raw materials for synthesizing new DNA segments and the cofactors that repair enzymes need to function. Let's now synthesize this into a practical dietary pattern that maximizes DNA repair. Here are evidence-based daily recommendations. 2 to 3 servings of leafy greens to ensure adequate folate intake. 1 to 2 servings of cruciferous vegetables for sulforaphane. 2 to 3 servings of berries for their high antioxidant content. 2 to 3 servings of fatty fish per week for omega-3 fatty acids. A daily handful of nuts and seeds for vitamin E, zinc, and selenium. Another important question: what about supplements versus whole foods? The scientific consensus is clear. Whole foods are superior to isolated supplements for several reasons. First, whole foods contain hundreds of bioactive compounds that work synergistically. You can't replicate this complexity in a pill. Second, nutrients in whole foods often have better bioavailability because the food matrix aids absorption. Third, whole foods provide fiber and other beneficial components that supplements lack. Let's discuss an often overlooked factor: timing of nutrient intake. Research shows that DNA repair mechanisms follow circadian rhythms. They're most active during certain times of day. Peak repair activity occurs during sleep and morning hours. This suggests that nutrient timing might matter. Consider consuming antioxidant-rich foods in morning meals to support peak repair periods. Here's a practical question: how quickly do dietary changes affect DNA repair capacity? Research indicates that measurable improvements in DNA repair markers occur within 2 to 4 weeks of dietary intervention. However, full optimization of repair systems may take 3 to 6 months of consistent dietary patterns. Let's explore the epigenetic dimension of nutrition and DNA repair. Your diet affects DNA methylation patterns, which are epigenetic modifications that regulate gene expression. Methyl donors like folate, vitamin B12, choline, and betaine are essential for proper methylation. The best food sources of methyl donors are: eggs are rich in choline, providing about 150 milligrams per egg. Beets are rich in betaine, which supports methylation cycles. Liver is exceptionally rich in both B12 and folate. There's also an important connection between detoxification and DNA repair. Phase 2 detoxification enzymes neutralize carcinogens before they can damage DNA. Cruciferous vegetables induce these phase 2 enzymes through compounds like sulforaphane and indole-3-carbinol. The pathway is cruciferous vegetables lead to phase 2 enzyme induction, which leads to detoxification of carcinogens. Now let's discuss which foods to minimize for optimal DNA repair. Avoid processed meats containing nitrites, which can form DNA-damaging N-nitroso compounds. Minimize charred or heavily grilled meats, which contain heterocyclic amines that damage DNA. Limit alcohol consumption. Ethanol metabolism produces acetaldehyde, which directly damages DNA and impairs repair mechanisms. Avoid trans fats and oxidized oils, which promote oxidative stress and inflammation. Let's discuss an interesting phenomenon: caloric restriction and DNA repair. Research shows that moderate caloric restriction, about 20 to 30% reduction, enhances DNA repair mechanisms. The mechanism involves activation of AMPK and sirtuins, which upregulate repair pathways. Practical application. Intermittent fasting protocols may provide similar benefits without chronic restriction. Let me give you a complete daily menu that optimizes DNA repair based on everything we've discussed. Breakfast: spinach omelet with wild salmon and mixed berries. This provides folate, omega-3s, and antioxidants. Lunch: kale salad with walnuts, steamed broccoli and citrus vinaigrette. This provides sulforaphane, vitamin E, omega-3s, and vitamin C. Afternoon snack: 2 Brazil nuts and a small piece of dark chocolate. This provides selenium and resveratrol. Dinner: grilled fish with roasted Brussels sprouts and sweet potato. This provides omega-3s, sulforaphane, and beta-carotene. Let's take a long-term perspective on DNA repair and nutrition. DNA damage accumulates over decades. The goal is to minimize net damage by optimizing repair. Consistent dietary patterns are more important than occasional superfoods. Think of it as daily maintenance rather than emergency repair. The best time to start was 20 years ago. The second best time is now. Every meal is an opportunity to support your DNA repair systems. Let me provide a final comprehensive summary of the DNA repair diet. The foundation is a plant-rich, whole food diet with abundant vegetables, fruits, nuts, and seeds. Priority 1. Include cruciferous vegetables daily for sulforaphane. Priority 2. Consume colorful fruits and vegetables for diverse antioxidants. Priority 3. Include omega-3 rich foods 2 to 3 times weekly. Priority 4. Consume nuts and seeds daily for vitamin E, zinc, and selenium. Priority 5. Minimize processed foods, excessive alcohol, and charred meats. In conclusion, the relationship between nutrition and DNA repair is scientifically robust and clinically significant. Your dietary choices directly influence your genomic stability and long-term health outcomes. Optimal nutrition provides both the raw materials for DNA synthesis and the cofactors that repair enzymes require. The scientific evidence strongly supports whole foods over isolated supplements for comprehensive DNA protection. Remember, consistency over perfection. Sustainable dietary patterns matter more than perfect adherence to every recommendation.